This diagram illustrates the classic Watson-Crick DNA double helix from 1953. It presents a uniform, static, right-handed ladder. Inside a living cell, this classical view is an artifact. DNA does not exist as a monotonous, uniform cylinder. It is a dynamic molecule. Depending on its chemical environment and its exact sequence, its local three-dimensional topography warps, bends, and shifts. Every local twist, the depth of its grooves, and the flexibility of its backbone are dictated by the linear order of its 4 nucleotides. To understand how DNA functions as the genetic material, we must move past the rigid textbook model and examine the microgeometry of its atomic bonds. These chemical structures display the fundamental building blocks of this geometry, the heterocyclic aromatic bases. They fall into 2 distinct classes: the double-ring purines, adenine and guanine, and the single-ring pyrimidines, thymine and cytosine. Analytically, bases attach to deoxyribose sugars, linked by phosphodiester bonds from the 5' to 3' carbons, creating the structural backbone. Between the antiparallel strands, strict rules apply. Adenine pairs with thymine via 2 hydrogen bonds, guanine with cytosine via 3. Beneath these horizontal connections, the adjacent parallel base pairs stack on top of one another. Hydrophobic van der Waals forces between these stacked rings stabilize the interior of the helix. While the pairing rules of AT and GC are immutable, the spatial orientation of these bonds creates a framework for significant structural variation. The primary source of this variation lies in the sugar moiety. The deoxyribose ring is one of the most dynamic parts of the DNA molecule. This diagram illustrates the sugar pucker concept. Because of the bond angles between its 5 atoms, the carbon ring is not flat. It bends into an envelope shape, where one or more atoms pucker out of the main plane. The 2 primary structural states are defined by which carbon atom twists upward. In the C2' endo conformation, the carbon at the second position pushes up. In the C3' endo conformation, the third carbon twists out of the plane. The second variable is propeller twist. Paired bases do not sit perfectly flat like the rungs of a ladder. They rotate on their longitudinal axis relative to one another. These microatomic shifts, the sugar pucker and the propeller twist, act as the physical switches that cause the entire DNA polymer to bend and morph into distinct helical forms. Under standard, highly hydrated physiological conditions inside a cell, DNA adopts its most common configuration, bDNA. At the atomic level, bDNA utilizes the C2' endo sugar pucker. Base pairs exhibit a positive 16-degree propeller twist, optimizing stacking interactions. These microparameters dictate the helix's macrogeometry. bDNA makes one right-handed turn every 10.5 base pairs, creating a 36 angstrom helical pitch. This geometry yields a wide major groove and a narrow minor groove. This creates an optimal balance of stability while allowing cellular proteins to read the genetic sequence without unwinding the helix. If the environment changes and water is removed, the DNA molecule structurally reacts, compressing into a shorter, wider configuration known as aDNA. This compression follows a precise atomic shift. The sugar pucker transitions to C3' endo. Propeller twist increases to positive 18 degrees, aligning hydrogen bonds into a stronger, linear configuration. The widened helix packs 11 base pairs per turn, forming a deep major groove. aDNA demonstrates how environmental stress directly dictates molecular geometry. This compressed structural form is also the native state adopted by RNA-DNA hybrids during gene transcription. Under specific localized sequences, the molecule can undergo a structural anomaly that reverses the spin of the double helix. This is zDNA. It is a left-handed helix, entirely reversing the standard right-handed spiral seen in the A and B forms. This inversion occurs in regions with alternating purine and pyrimidine bases. These sequences force the glycosyl bonds to adopt alternating syn and anti conformations. Because bonds alternate directions, the phosphate backbone cannot maintain a smooth curve. It forms a rigid, stretched zigzag pattern. Unlike the 16 or 18-degree twists of other forms, zDNA features 0 propeller twist. Its base pairs sit rigidly flat. zDNA proves the double helix is not a fixed track. It is a responsive polymer, capable of localized radical inversions to relieve torsional stress generated during active transcription. When we expand our view beyond single base pairs, we reach the concept of the dinucleotide step. This is where the specific pairing of adjacent bases dictates the local twist and slide of the helix. An adenine-guanine step possesses a different stacking angle than a guanine-cytosine step. These distinct angles cause localized bends in the overall sugar-phosphate backbone. These sequence-dependent bends allow the polymer to fold and wrap precisely around histone proteins. This localized flexibility is required to generate the massive, dense supercoils that fit nearly 2 meters of genetic material into a microscopic nucleus. The information in the genetic code is stored within the linear chemical sequence and the resulting three-dimensional architecture. The monotonous helix is a pedagogical myth. DNA is a data-dense polymer whose topography responds to every chemical and sequence-based signal.